Hello, my name is Zuha Beg, and my project is on the effect of carbon dioxide on plant transpiration. So I did some research regarding the process of transpiration, which is how water vapor is released through the stomata of plants, through the leaves. And the stomata are pore-like structures found on the epidermis or outermost surface of the plants. And they may open or close due to internal or environmental cues. And this allows the water to either exit or it also allows oxygen, which is a byproduct of photosynthesis, to exit, and also allows carbon dioxide to be taken in, which is a reactant in order to undergo photosynthesis and produce glucose for the plant. And there are also many environmental factors that affect transpiration, such as humidity, temperature, light, and wind, other factors as such. And the stomata are composed of two guard cells, which take environmental cues, and they have receptors that take in these signals and perform based on those signals. The history of my experiment, I found that previous experiments have found that environmental pollutants such as dust deposition negatively affect photosynthesis because they block the stomata. And I also found that environmental gaseous pollutants such as silicon dioxide and ozone may affect transpiration by causing stomatal closure or opening. And it also affects the carbon dioxide sensors within the stomata, and this may also have an effect on how transpiration occurs, and this is directly related to my own experiment. I feel that my experiment is very important because transpiration directly affects the environmental humidity of our atmosphere, and it's very important because humans are constantly polluting the environment, and it is important to understand how this may affect the way plants function, and they are a vital part of our ecosystems. I hypothesize that if basil plants have a lower rate of transpiration with an increasing amount of carbon dioxide available, then the plants with a greater concentration of baking soda will have a smaller amount of milliliters of water lost than those with the lower concentration of baking soda. These are the materials that I used, and basically my procedure was that I took 4 plants, placed them each in a jar, and I gave each a different concentration of baking soda. One of them had 0 grams of baking soda, and I allowed them to sit by a window and let them sit for 24 hours, after which I recorded their mass again. And the mass lost was the amount of water that was transpired. And these were my results. I found that the plants with a higher amount of concentration of baking soda had less water transpired, and the one with the least amount of baking soda, which is 0 grams, had the most amount of water transpired. And some discrepancies in my data may have come from the baking soda not being able to fully dissociate into carbon dioxide gas, because adding heat may not have fully sped up this chemical process. So I concluded that the results proved my hypothesis to be correct, and with increasing concentrations of baking soda and increasing concentrations of carbon dioxide, the amount of water transpired by the plants decreased. And carbon dioxide essentially did decrease stomatal conductance, and it decreased the amount of water that was able to leave through the stomata of the plants. I would like to acknowledge Mrs. Soria, my science research teacher, for helping me through this process. And these are my works cited, and that is all. Thank you.